Good morning, sir. So, Navdesh here. Where highly welcomes you on behalf of Student Research Lab, Ireland DPU Pharmacy. I am honored to have you as my guest, first guest, especially for the podcast we are going to shoot together for. So, sir, I like to ask you, sir, how would you describe your life journey so far? You have reached a stage where many look up to you as a motivation, as a speaker, as you speak today. What key experience of discussions or the idea you explore in the work of research help you and motivate you to do?
Mm. It is totally interest and a challenge which was important for me to come up. And hardworking also. Those days there were not the facilities like presently available. So plenty facilities are very fast, but as I can see, they are artificial. Those days they used to be developed through brain, hardworking interest, and the way our seniors used to make us to work. So that's what made me to learn a lot of things through challenges and also develop new technology, new molecules, disprove the mechanism of action of certain drugs proposed by foreign companies. Sometimes discouragement to me during my study and later on in the research field helped me to improve myself and take up the responsibility.
So my next question would be, what is your message for the new students who are just starting their college in pharmacy to elevate their interest in research and initiate at early ages as either do this immense work? How would have you started? Yeah, how would you have, as a fresher, start research today?
First of all, you have to look into the before starting new adventurous work. Look at the reputation of the college. There are so many pharmacy colleges in the Maharashtra, also in India. You have to choose. Frankly, to tell you, undergraduate students going for post graduation or doctorate must analyze, understand the contribution of a person to whom you want to join for research. Facilities availability, freedom of availability. This is what I was looking for. One example, I was in a pharmacy college. There was nothing. I was made to teach a diploma class. It helped me. Still, then I used to go to an reputed institution to carry out the research, mainly because I had the interest for research and to tell the public that there is something different going to happen in the future. Not the same what he had done, what she had done, what we used to do. So hence, I am rated something differently as in computer readable. So this is what was my motive. So today it is like that. If there is a person like that, CADD, drug discovery, drug design, people know there is VM Kulkarni. Intuition, interest, motivation. And then intuition is a very big word. It has to come from different people also. Certain, not to name that thing. When people, for example, when I returned from Germany, I joined a pharmacy college. People knew, industry people knew that such and such person has joined a pharmacy college in Mumbai, Bombay College of Pharmacy. Immediately, 11 research projects for industry came to me in the synthetic, also in the formulation. I'm not a formulation scientist. I did formulation also. It's only interest to take the challenge. But sorry to say, I changed the college, went to another institute where I have the freedom to do the work is ICT.
And my next question would be, could you share the story when you challenged the UK scientist on mechanism of action of beta blockers? How did you manage to do this with a limited resource at that time available in India?
At that time, I was not a PhD student, but a postdoctoral visiting scientist at the Tata Institute of Fundamental Research in Kulkarni, Mumbai. Tata Institute of Fundamental Research is not for drug research. It's for, as I said, it's a nuclear physics, astrophysics, biophysics, mathematics, many things. It's a highly reputed institution. I was invited to join there. Under whom I joined was not a drug chemist or any chemist or anything. Since I studied in medicinal chemistry, I always used to see the medicines in chemistry. We used to read the books, journals. One paper in the Journal of Medicinal Chemistry was published by a scientist for Imperial Chemical Industries, UK. And he had determined the structure of beta blockers. Example, aryl oxypropianolamines. Aryl oxypropianolamine, propranolol, atenolol, alprenol, oxprenol, timolol, acetylol, etc. He proposed the mechanism of action of these aryl oxypropanols, which I disproved. He had determined the molecular conformation of aryl oxypropanol amines using non-polar carbon tetrachloride, chloroform, and determined the molecular conformation of the aryl O CH2 CH OH NH CH like the aryl oxypropanol amine. It is a elongated structure. And he proposed by determining their molecular conformation that in the body this undergoes a rigid conformation. Body, if the drugs are rigid and they undergo the rigid means strained. If they are strained molecules and if such drugs are administered, they don't show the activity. When I studied his paper, I told my mentor, sir, this paper is wrong. What are you doing? He told me, sir, they have determined the molecular conformation using a non-polar solvent, which a body doesn't contain benzene or non-polar solvent. It cannot undergo rigid conformation in the body. So his proposition is wrong. Then what do you want to do? Determine the molecular conformation using the physiological pH solution, which is resembling the biodium, like GIT pH, stomach pH, blood pH. Determine the molecular conformation of the hydroxypropanols in a different pH using NMR. That time, NMR was 100 milliards a day. It gives you how the molecule orients Gauss conformation, trans conformation, etc. When I studied the atenolol, propranolol, adiloxypropyl, etc., by NMR and determining their coupling constants, population of the conformer, he interpreted the NMR. My guy, I proposed the mechanism, sir. These molecules undergo trans conformation in a straight way, then bind to the receptor to give result. This was published in International Journal of Quantum Biology. And my mentor was attending the conference there. And that person who had proposed rigid conformation, he came to Arya Kulkarni. He came and met him, Arya Kulkarni. You have disproved my theory. I congratulate you. He said, I'm not Kulkarni, he is my student. So, hence, that was the big thing like that we proposed. On that basis, after confirmation, ICICI company discovered acetylbutanol, oxprenol. Yes. Mm. But India was not geared up for that at that time for new drug discovery.
Sir, in one of your interview, you have mentioned that when you are coming from Chennai, you have malaria, and within a train journey. Sir, I've seen your interviews. For preparation of the questionnaire, I've seen your interviews, and I've also read your book in my provenance in my 12th when I'm doing bioinformatics.
So, okay.
So in one of your interviews, you mentioned working in Chennai, contacting Maria, and writing a book while you are traveling in a train. So, sir, I want to know what fascinates you about the process of learning and then sharing knowledge while this difficult challenge.
I enjoy sharing my knowledge. I enjoy teaching the students. I enjoy motivating the students. It's not an industry. College students are not industry. We have to make them for industry. That is what you have to teach in that way. So that's why wherever I go, without any education, I tell everything. It is a good teacher. Teacher who keeps something inside him is not a good teacher. He must tell. All over India, I have taught like this from Kanyakumari, Jammu Kashmir down to Punjab. Also lectured in Madurai and things like. In East asked everything, introduced to the subject and taught them with the chalk and blackboard. What it made me, how I developed the memory. Is this what watching?
Yes, sir.
When I was in ICT, joined as a professor in medicine and chemistry, and became head of the Department of Pharmacy and chairman of pharmacy. I used to work with my hands in the laboratory, being head everything, lot of administration. After everybody has gone, students only I used to be here. I was preparing myself some compounds and things for the do it right. I developed a process for a drug called alprazolam. Structure? Not know? No. In 4 steps only. A company in Chennai called Ranipet, Malari Drugs and Pharmaceuticals Limited, in Ranipet, Chennai. They came to that here is one Kulkarni in ICT has developed the process. At that time, that drug was costing 4 lakhs rupees crore. And this is important, don't put it. They came to me at ICT, sir, we are interested in having the process technology, lab scale only for alprazolam, which you have done. Can you share with us? Yes, give all the facilities. Chemicals only needed, flask, condenser, and such things. Nothing, and sterile without. They managed everything and gave to me. I went to Ranipet. Good. good treatment. I showed them alprazolam of 100 grams, and they paid me 1 kilo price. This is how chemistry gives money also. Chemistry brings reputation also. Initially, you may suffer. There is money in chemistry. So, when I was demonstrating alprazolam synthesis in the laboratory of Mongolian drugs, there were a lot of mosquitoes. And they bit me like anything. So, second day, I fell sick with malaria. Last stage of the alprazolam. I had told them when hydrogen sulfide starts eliminating from the reaction, think that the product is clean. This much I had told, and I slept with it. I slept with the malaria. Jumping. One should experience malaria. And if not treated immediately, and if the parasite, plasmodium, or falciparum, if it travels to the brain, finish.
Yes.
So it is required to be administered every 4 hours. 3 hours, 4 hours of 10 milligram. So third administration of 30 milligram went into the body. So jumping stopped because it has to absorb and distribute. So that distribute in the blood. So that the parasites kill. Ultimate aim is to control the temperature rising. So had to be administered. Doctor does not know the side effects of toxic effects of flora. Maybe knowing 30 milligrams when I took. God knows what happened. So many things started coming into my mind, particularly chemistry. They put me in the train. Drug design, drug discovery. So that thing started coming in my mind like anything. I did not write. I wrote in my brain only. When I arrived at Bombay, I told my wife, I don't know what is happening at that when my marriage took place. All that. काय झाले तुम्हाला असं काय बोलावं लागलं अरे काही नाही असं असंच आहे काय नाही आय एम रायटिंग आय स्टार्टेड रायटिंग द बुक ड्रग डिझाईन हाऊ दिस हॅपन as a side effect, toxic effect to that in persons, some persons, or in a number of persons, I read about it, that it enhances the CNS system or depresses the CNS system. If it depresses, patient will undergo suicidal tendency. He will feel like falling down. This is suicidal chloroquine effect. In some other cases, it gives enhancement, memory development. It happened fortunately in my case, the memory enhancement took place. So that is CNS effect, thanks to chloroquine. That's what how I remember many structures, many dogs. So I speak so much, I don't do you understand? Continuously flows. Names of the drugs continuously flows. Interesting. Thanks to Porgan.
Sir, my next question would be, what methodology you use while learning new things? What methodology, what process you use when you learning new things? Sir, and also I want to know how you start learning anything new, how you choose the topic.
No, not research work.
Yes.
See, much experience with me this time. And mind has read a lot. It is my 46 years in teaching and research, 44 research work, 44 years. So much so traveled out of India, Germany, 3 years I was in Germany, so many Netherlands, Holland, this thing, and America, many places. First time I was invited to give a talk in Gordon Conference. When in India had a Gordon Conference in medicinal chemistry, Gordon Conference in drugs and things like that. And then I traveled almost Australia, Japan. All this travel interaction with the people made me to get, why should I not do the work they are doing? Industry people, why you want to do the synthesis of ciprofloxacin? How did this was discovered? Why you are not telling? I asked the managing director. So with this type of knowledge and the students' interest, do them within the available facilities, what the student can do, simple thing at the post-graduation level. In ICT, because of facilities and things like that, very tedious synthesis, very even because hydrogenation, pressure reactions. So these are not there in the instance. Simple in the college life and simple in class content that is also not available sometimes. Good idea. Yeah, I thought basic facilities. That's how I think of, because I still read, browse the journals, see the internet. Why? And also I try to find out the flaws in the drug. Why this drug is toxic? How do you improve the toxicity? That way I think and try to give, but present day, it has become difficult in the setup available. So this is how I assign the research project in simple one. For example, right now one student is doing anti-cancer sulfonylurea as a one-step synthesis only. That one-step synthesis only. 4-methyl phenyl sulfonyl isocyanate reacted at room temperature with differently substituted anilines to get 4-methyl or 4-substituted phenyl sulfonyl uridophenyls as anti-cancer, and they are showing anti-cancer activity for MCF7 and MTT7, breast cancer. Thus, how to make a simple chemistry is my interest. And now, enough for me. Enough for me, you understand?
Yeah.
खूप झालं. If anybody student is interested, okay, I still like to continue. I have a new offer in Bangalore. I'm living from here.
So my next question and final question is that if you could restart your life, what, if anything, would you do differently? If you can restart your life from the beginning,
Restart my life. Only God has to restart.
if you had your chance, if you had your chance to restart your life, what you would do differently? Or if you're not a scholar, what you have choose as your profession?
Something innovative work I would like, which others foreign countries have not done. I want to do something very thrilling, interesting, so that it is useful to the manpower, suffering mankind. Why I returned from Germany after 3 years, that India, there are a lot of leprosy patients. I want to develop a drug for the treatment of leprosy. So this is that let our country, otherwise I would have remained there only or gone out of India to some other foreign countries as a present day students are settling there. That is not me. My children also, they are also PhD from America. They also came back to India. They are big scientists here in India. So I told him, First, your country. You use your knowledge, irrespective of so many problems. Get adjusted to the environment. Do contribution to your industry is my interest. Some minimal 38 students or 42 students are in America. They don't want to come back. But you can get anything you think in America. You won't get parents here. Everything, freedom is there like anything. Lots of facilities are there. Everything is there. Where that here also, you show your expertise. Government is ready to give money like anything. That innovation, something different is required here. If you show that you put a proposal, if you put a project, something very different, they will finance. But some reputation, many things are there. It takes time. Students have to be motivated about that. Students have to show their interest. I also come up with as a student only.
Yes. Yes. Thank you for being my guest.